right? I will start with the first lesson, Pute, right? Grade 10, Unit 1, Chemical Basis of Life, right? The first lesson. Right? Most probably the first MCQ questions for the O-level paper also based on this lesson, right? So it is very important to go through this lesson, Pute, right? Right, in this lesson, mainly we discuss about the living matter and mainly the animals and the plants and how their body, how the body of the living organisms made up of, what are the components and what is the structure of those components, what is the significance, how we can identify those components, like that, right? Living matter, right? Here living matter means both the animals and plants, Pute, right? All the living organisms, right? So when we consider all the living organisms or the living matter, right, mainly there are we can identify the most common elements in the living matter, right? Most common elements that we can find in the living matter, right? What are the most common elements, Pute? Yes, carbon, hydrogen, oxygen, and nitrogen, right? These are the most common elements. Other than these elements, there are some other essential elements that we can find in the living matter, right? Essential elements. Right? What are the essential elements? Sulfur is there, phosphorus, right? Sodium, potassium, chlorine, right? And iron is there, right? And calcium, magnesium, like that. There are some other essential elements that we can find in the living organisms, right? And when we consider these elements, by combining these elements, right? By combining these elements, chemical compounds are formed, right? Chemical compounds are formed. With the combination of these elements, maybe the common elements or other essential elements, chemical compounds are formed, right? We can divide or we can categorize these chemical compounds, Pute, into two main groups. I think that you can remember. Yes, what are the main two groups of chemical compounds? Yes, organic compounds and inorganic compounds. Organic compounds and inorganic compounds. By combining these elements, the more common elements or other essential elements, it forms the chemical compounds, right? Our body, the whole body of the living matter consists of these chemical compounds, maybe the organic or inorganic compounds, Pute, right? So these organic compounds we get as the biological molecules, right? Biological molecules. In the first lesson, we discussed about four types of biological molecules, Pute, right? In the first lesson, we discussed about the main four biological molecules, right? I think that you can remember. What are the main biological compounds? Carbohydrates, yes, proteins, lipids, carbohydrates, proteins, lipids, and nucleic acid. Right? So in this lesson, we discuss about these four biological molecules, Pute, right? How they are formed, right? What are the functions of these biological molecules and how we can identify these four types of biological molecules, and what are the examples for these biological molecules. right? This is the basis of this lesson, Pute, right? If you can make the note with me, it is okay, right? If you can make a rough note when I am writing, right, then you can make the note. Or after the class, again, you can go through the recording and you can add your own things also to the note, Pute, right? And if you did not study the lesson, you can study the lesson and go through the recording, watch the recording, and with my short note, add your ones and make another short note as you wish, right? I am only giving the only the structure, right, Pute? Right? So this is the basis in the first lesson, right? Then I will erase this part, right? I will erase this part and we will move to the next section of the first lesson, Pute, right? Right. Then in detail, we have to go through about the biological molecules, Pute, because there's a vast section in of the about the biological molecules, right? Biological molecules. Right. So I told you in this lesson, we have to discuss about 4 biological molecules, Pute, right? Then we divide this one into 4 sections, right? This one you can do as you wish, right? If you write one by one, carbohydrate, then protein, then lipids, like that, then it is okay, right? Carbohydrates, proteins, right? Lipids and nucleic acid, right? Then about carbohydrates, Pute, right? When we consider carbohydrate, yes, what are the main elements that consist in carbohydrate? Carbon, hydrogen, and oxygen, right? Major elements in carbohydrates: carbon, hydrogen, and oxygen, right? Then you have to know the examples for carbohydrates, or what are the types of carbohydrates. When we consider that one, right, I think that you can remember there are, right here, we discuss about the 3 types of carbohydrates, right? Types of carbohydrates. Yes, I write in short form: monosaccharides, yes, disaccharides and polysaccharides. Monosaccharides, disaccharides and polysaccharides. Mono means one, Pute, right? So the monosaccharide is the building unit of carbohydrates. Monosaccharides are the building units of carbohydrates, right? Then we will write the examples for these 3. Yes, what are the types of monosaccharides? Yes, glucose, fructose, and galactose. Glucose, fructose, and galactose, right? Then what are disaccharides? By the combination of 2 monosaccharides, it form a disaccharide with releasing a water molecule, right? But then what are the disaccharides here? There are 3 disaccharides given in your book, Pute, right? Maltose, sucrose, and lactose. Right? By the combination of two glucose molecules, it forms a one maltose molecule, one disaccharide, right? By the combination of glucose and fructose, it forms a sucrose molecule, Pute. By the combination of one galactose and glucose, it forms a lactose molecule, right? In these three instances, when combining two maltose molecules, it releases one water molecule, right? Then what are the examples for polysaccharides? Yes, cellulose, only we can find within the plant cells, starch, and glycogen. Right? So these are the three types of carbohydrates, Pute. Right? Monosaccharides, disaccharides, and polysaccharides, and examples for these three. Right? Then the other one, right? You have to remember the functions of carbohydrates, Pute. That is another section. Right? I'll write here. Functions of carbohydrate. You can use some other color pens, right? If you have. Right? Functions of carbohydrates. In short form, you can write the functions of carbohydrates. Yes. As a source of energy. Right? It is important as a source of energy. Yes. And it is a structural component. Right? Structural component. Right? And it is a component in nucleic acid. Component in nucleic acid. Right? Like that, you can write the functions, Pute. Right? And there is another thing about carbohydrate, that is the test to identify carbohydrate. Right? How we can identify carbohydrates. Right? Here there is a test for identify starch. Right? And to identify glucose. Right? When I am discussing the practical section, Pute, in the biology part, right, I will explain these practicals, right, how we can identify these things, right. So those are the main things that we have to focus on carbohydrates, right. Then we will move to the proteins, right. When we consider the component elements, right, other than carbon, hydrogen, and oxygen, nitrogen is there in proteins, Pute. Carbon, hydrogen, oxygen, and nitrogen is there in proteins. Right? Then here also, we have to remember the functions. Right? Functions. Right? The functions of protons. It is also an energy source. Right? All the things are given in your book. Right? And structural component. That means proteins participate to make the structures, right? Structural component. And, right, you know, right, as enzymes, all the enzymes are proteins and all the hormones are proteins. Enzymes and hormones, and as antibodies, right, as antibodies. Right, you have to remember this one, Pute. All the enzymes are proteins, but not all the proteins are not enzymes. Right, remember this one. All the proteins are not enzymes, but all the enzymes are proteins. Right, and the test to identify proteins. right? The test to identify proteins, there is only one, that is biuret test, right? Biuret test. Right? We use the biuret test to identify proteins, Pute, right? They are at when we do the biuret test to the protein solution, we add excess amount of sodium hydroxide, right? First, we have to add sodium hydroxide, Pute, excess amount of sodium hydroxide and few drops of copper sulfate. It is very important to remember this one, right? When we do the biuret test for the protein solution, we add excess amount of sodium hydroxide, then few drops of copper sulfate, right? Then we can get the color change, right? That one also we discuss in the practical section, right? Then we move to the lipid section, Pute, right? About the lipids, right? In lipids also, as the component elements, same as in carbohydrates, right? They are the major elements, carbon, hydrogen, and oxygen. Carbon, hydrogen, and oxygen is there, right? Right? Then when we discuss how the lipids are formed, they form by the combination of fatty acids and glycerol, right? We can write that one also. Lipids are formed by the combination of fatty acids and glycerol, right? Fatty acids and glycerol. Fatty acids and glycerol. By the combination of fatty acids and glycerol, it forms lipids, Pute, right? Right? Then here also, right, to identify the lipids, we use a test, right? That is Sudan 3 test. Right, it forms the pink color, or we can say red color globules are formed when we add Sudan 3 region to the lipid solution, right? Sudan 3, right? And here also you have to remember the functions, Pute, right? Functions of lipids, right? They are also as an energy source, right? Same as carbohydrate. After finishing the carbohydrate, right? Normally it use lipids to supply energy, right? And it also acts as a structural component, mainly in the plasma membrane, or we say the cell membrane. Structural component, right? And there is another special thing to maintain body temperature, right? To maintain body temperature, lipids help for that one, right? And there are some other functions given in the textbook, Pute, right? It is better to go through them, right? That is about the lipid. Then we will move to the nucleic acid part, right? When we consider the consisting elements in nucleic acid, right? That is carbon, hydrogen, oxygen, and nitrogen is there, and phosphorus also there, right? Carbon, nitrogen, oxygen, carbon, hydrogen, oxygen, nitrogen, and phosphorus is there as the component elements in nucleic acid, right? And as the building unit of the nucleic acid, Pute, right, we get the nucleotide, right? Nucleotide. Nucleotide. Right? Nucleotide. When we consider one nucleotide, there are main three parts in one nucleotide, right? Major three parts in, or major three components in one nucleotide. What are the three major parts? Yes, the nitrogenous base is there. Right? Nitrogenous base part is there, and the pentose sugar group is there. And the phosphate group is there. And the phosphate group is there. Right? So, these three are the main three components in one nucleotide, Pute. Right? So, when we consider the nitrogenous base and the pentose sugar, right? Nitrogenous bases and the pentose sugar, mainly there are two types of nucleotides. There are two types of nucleotides, or we can say there are two types of nucleic acids, Pute. Right? What are the two types of nucleic acid? Yes, DNA and RNA. DNA and RNA. Right? In DNA and RNA, Pute, pentose sugar is different and the nitrogenous bases are different. Right? Only one base is different, other three are same. Right? But you have to remember the pentose sugar group is different in the two nucleic acids. Right? Here, the building unit is in DNA deoxyribonucleic acid. Deoxyribonucleic acid, right? And the building unit is deoxyribonucleotide. Deoxyribonucleotide. By combining deoxyribonucleotides, it forms deoxyribonucleic acid, right? Nucleic acid means something like the polymer, and nucleotide is the monomer, right? You know that the monomer is the building unit to form the polymers, right? So, by combining many nucleotides, it forms nucleic acids, Pute, right? So, by combining deoxyribonucleotide, it forms deoxyribonucleic acid or DNA, right? So, in DNA, the pentose sugar is deoxyribose. Here in RNA means ribonucleic acid. Ribonucleic acid, right? That is the nucleic acid. And the nucleotide is ribonucleotide. Ribonucleotide, right? Ribonucleotide, right? So those are the main two nucleic acids, DNA and RNA, right? And separately, you have to remember the functions of DNA and functions of RNA, right? What is the function of DNA, right? It transfers genetic material. Transferring genetic materials from one generation to next generation, right? That is the main function. And there is another one, storing the genetic information, that is also in DNA, right? Storing genetic information and transferring genetic materials or genetic information from one generation to next generation, right? That is the function of DNA. Then what is the function of RNA, Pute? Yes, protein synthesis, right? Protein synthesis. That is the main function of RNA. There are 3 types of RNA, mRNA, tRNA, and rRNA, right? All these types of RNA help for the synthesis of protein, Pute, right? So, if you ask to write the functions of nucleic acid, you can write these both, right? Right? They are as common, you can write the functions, Pute, right? What are the functions? Yes, storing genetic information, right? And transferring genetic materials or genetic information from one generation to next generation, and protein synthesis, right? As common, the functions of nucleic acid, you can write all these, right? Or if you have asked separately, what are the functions of DNA and what are the functions of RNA? There you have to write them separately. There, for DNA, you can write about that one. Storing genetic information, that is one thing, and transferring genetic materials from one generation to next generation, right? And the functions of RNA, protein synthesis, right? Those are the things, Pute, about the biological molecules, right? When you make the note, right, you can write more clear than this one, right? Manage the space. You can use a A4 paper or a foolscap paper, and you can gather all the things about biological molecules in one sheet, right? Then it is very easy. And when you are studying, Pute, right, you can go through in this way, that the biological molecules, protein, what are the components, elements, right? What are the functions? Then what is the test that we can use? Yes, that is biuret test. How we can do the test, right? What we add, what are the regions we are adding? Then what is the observation given? Like that, Pute, right? When you go through this part, when we consider the protein section, right? Studying means, right, you can speak up, right? You can talk with you, right? Proteins, these are the components, elements, and these are the functions. This is the test. What are the things that we add? What is the observation we get? Like that, right? You can practice, Pute. Then it is very easy, right? You are writing, then the message sent to the brain, right? You are speaking, then from hearing again the message sent to the brain, right? Then from the eyes, you look at this one, no, your short note, then from the eyes also the message sent to the brain, right? The same thing, right? The same thing sent to the brain in 3 ways, right? From the eyes, from the ears, right? And from the hand, right? Then it is very easy to remember these things, not like this way, right? Go through the lesson in this way, right? It is very hard to remember, right? If you have a good memory power, then it is okay. But when you practice in this way, Pute, right? That is why I told you to use the color pens, right? It is very easy to remember the things with colors, right? Because our brain is like to remember the things with colors, Pute, right? So don't use many more colors, but that you can use in this way, Pute, right? For the main topics, you can use one color, right? For the subtopics like this, you can use another color, right? To highlight the things, you can use another color, right? Like that, you can make a sketch in your brain, right? When you are making the short notes, right, very first, you can imagine or you can clarify those things. For this one, I use this. For this one, I use this. Like that, right? You can manage the things. Then with the color also, you get the idea, right? When you are remembering, you can remember that one. Yes, I have written in red color. That means it must be a main topic, like that, right? You can get the idea, right? So when you make the short notes, Pute, right, use all these methods, right? Then it is very easy to remember, right? I think you got my idea, right? Then This is about the biological molecules. I think you can make a note better than this one, right? Right? Then we will move to the next one, Pute. Right? So our next biology lesson is the sixth lesson, Pute. Right? Sixth lesson, that is about the plant cell and animal cell functions and the structure of plant and animal cell. Right? Functions and the structure of plant cell and the animal cell. Right? You know that the cell is the main or the basic structural unit and the functional unit of the living organisms, right? In both animals and plants, right? The cell is the basic unit, Pute, right? So, in this lesson, first you are given that how the basic unit, that means the cell is observed by the Robert Hooke, right? By the Robert Hooke, and he gives the name as the cell, right? The basic structure or the basic unit named as the cell, right? And the cell theory is given, Pute. You have to remember the three scientists who put forward this idea. Yes, Matthias Leiden, Theodore Schwann, and the Rudolf Virchow, right? With the correct spellings, you have to remember the three scientists. And what is mentioned in the cell theory? Yes, what is mentioned in the cell theory? The structural and the functional unit is the cell, and all organisms, maybe the unicellular or multicellular, all the organisms are made up of cells, right? With one or more cells. If they are unicellular, the whole body is one cell. If they are multicellular, there are many cells in the body, like us, right? And new cells are formed by the existing cells, right? So those are the three facts given in the cell theory, right? I'm not going to explain those things, right? You can remember these things, right? And how we can observe the cells. For the to observe the animal cells, we use the cheek cells, right? We make the slide, and under the microscope, light microscope, we observe the animal cells, right? And to observe the plant cells, to get the idea about the plant cells, we can use the lower epidermis of beetle leaf or ohio or the onion peels, like that. To observe the plant cells, we can use those specimens, right? And the major thing is, Pute, right? To get the idea about the plant cell and the animal cell, right? Animal cell and the plant cell, the structure of the animal cell and the plant cell, right? For this one also, you can use different methods, right? Right? Animal cell and the plant cell. By drawing the figure, by drawing with the figure, you can make a comparison with make a comparison of the animal cell and plant cell, Pute, right? I am drawing only the rough sketch, right? The animal cell, right? The outer layer is the cell membrane, but when we consider the plant cell, the outer covering is the cell wall, right? When you draw, draw the clear diagram, right? Only you have to draw one diagram, no, right? So draw it clearly, right? When we consider the plant cell, there's a large vacuole in the center or in the middle, right? So the cytoplasm is, right, transferred to the periphery of the cell. But here in the animal cell, it is not like that one. The nucleus is there and we can find other organelles, right? And the special feature here is the central vacuole is there, and we can find another organelle, especially in the animal cell, sorry, plant cells, Pute, that is chloroplast, right? That is help for the photosynthesis or the where the photosynthesis is going on, right? So in the plant cell, cell wall is there, large central vacuole is there, and chloroplast are there, right? In the animal cell, the outer covering or the outer layer is the cell membrane. Here, the cell wall, next to the cell wall, cell membrane is there, right? And all the other organelles are same in both the animal cell and the plant cell. We can find all the other organelles except the chloroplast and the large central vacuole, right? So when we consider the differences, right, here in the animal cell, right, animal cell, cell wall absent, right? Here, cell wall present in the plant cell, right? And here in the animal cell, right, large vacuole is absent, right? Sometimes we can find small vacuoles, but here in the plant cell, large vacuole in the middle is present, right? Here in the animal cell, no chloroplast are there, chloroplast absent, but here in the plant cell, we can identify chloroplast, right? So those are the major differences between the animal cell and the plant cell, right? With the diagram, right, you can compare these things, Pute, right? In the animal cell, no cell wall, no large vacuole, and no chloroplast in animal cells, right? But in plant cells, there's a cell wall made up of cellulose. It combined with the first lesson, and there's a large central vacuole, and chloroplast are there. Chloroplast is the place where the photosynthesis is going on. Inside chloroplast, we can find the pigment chlorophyll, right? Chlorophyll that is help for the photosynthesis, right? So that is about the animal cell and the plant cell. Right? Then we have to know about the organelles, right? The organelles in the animal cell and the plant cell, right? Right, there you have to remember the figure of the organelles, Pute, right? So when you draw the organelles, draw the figure also and write down the functions, right? When we consider, right, here the commonly we can write all the organelles, right? When we consider this like this, the cell wall, right? Right, the cell wall. You can draw it like this, the cell wall, right? And you can write the function here, Pute, right? What is the function of the cell wall? The main function, right? It maintain the shape of the cell, right? Maintain the shape of the cell. Maintain the shape of the cell. Here cell means the plant cell, right? And it support for the cell, right? And it gives a protection for the cell, right? In that way, you can write the functions of the organelles, right? Then the cell membrane or the plasma membrane. All the things are given in your textbook, Pute. Only you have to go through it and make the note in this way, right? Then it is very easy to remember. When you are writing 2-3 times, right? Very easily you can remember those things, right? Cell membrane, right? Then the nucleus, right? The major organelle in all the animal and plant cells, right? Nucleus, right? And the mitochondria. Yes, mitochondria we consider as the very easily we have to remember the mitochondria. It is a double layer organelle, Pute, right? This is the outer layer or the outer membrane, and the inner membrane folded in this way, right? When you are given the diagram or the figure, very easily you can identify, right? The inner membrane folded in this way, right? The mitochondrion, right? Very easily you can identify. We consider the mitochondrion as the powerhouse of the cell, right? Powerhouse of the cell, because it produces energy, right? Mitochondrion, that is the function, producing energy. That is why we consider the mitochondria as the powerhouse of the cell, right? Producing energy by the aerobic respiration, producing energy, right? And the fifth one, when we consider the Golgi complex, if you are given the diagram, you have to identify, Pute, right? The Golgi complex. Some sacs are there, one over the other in this way, right? The Golgi complex, right? Some sacs are there and the vesicles are here, right? The Golgi complex. And the ribosomes, right? Ribosomes and the endoplasmic reticulum. Endoplasmic reticulum, I will write here as the sixth one, right? Endoplasmic reticulum in two ways, right? RER and SER. Yes, ER means, you know, endoplasmic reticulum, Pute. RER means rough endoplasmic reticulum. Rough endoplasmic reticulum. If the ribosomes attach with the endoplasmic reticulum, we name it as rough endoplasmic reticulum. If the ribosomes are attached, right? If the ribosomes are not attached, we name it as smooth endoplasmic reticulum, right? Smooth endoplasmic reticulum, right? So there are two types of ER or endoplasmic reticulum, RER and SER, Pute. RER and SER, right? Separately, you have to remember the functions of these two, right? RER and SER. In RER, the function is transporting proteins, transporting proteins within the cell, right? In SCR, synthesis of what? In SCR, synthesis of what? The lipids and steroids, right? Separately, write the functions of RER and SCR, right? One by one, you can write these things, right? And the vacuole in the, I will write here as the seventh one, right? The vacuole in the plant cells. It is also a cell organelle and it is helpful the maintain the water balance, right? Vacuole help for maintain the water balance, and all the pigments are dissolved in the sap of the vacuole. All the pigments dissolve there, so it support to make the shape of the cell, the vacuole, right? Right. In this way, one by one, you can write all the organelles puthe and make a diagram, right? Draw the figure, then it is easy to remember. Or if you are given the diagram, then it is very easy to identify and write the functions, right? And this one, right? Uh, right. So that is about the cell organelles. Right. And the diagram for the chloroplast is not given here, Pute, right? This in this way, right? Some sacs are there, and it is also a double layer membrane, right? In this way. Right? Chloroplast, right? And this is the mitochondria, right? Mostly you mix up these two, right? In mitochondrion, the inner membrane folded. But here in chloroplast, we can identify these sacs, right? In this way, right? This is the chloroplast, only we can find in the plant cells, and this is the mitochondrion, right? The powerhouse of the cell, right? Mitochondrion we can find in both the animal cells and plant cells, right? This is chloroplast, and this is mitochondria, right? You have to remember with the figure, Pute, right? So that is about cell organelles. Right? And there is another special thing here in this lesson, that is about cell division. Cell division. Right? Cell growth and cell division. What is cell growth, Pute? What do you mean by cell growth? Yes? Growth of a cell means that is the irreversible increase in the size of the cell or the dry mass. Irreversible increase in the size of the cell or the dry mass, right? That is how we define the term growth, right? And about the cell division, right? When we consider the cell division, right? Cell division, it takes place in two ways. Cell division. Yes. Can you remember? Yes. What are the two ways? Mitosis. Yes. Mitosis and meiosis. Mitosis and meiosis. Right? Mitosis and meiosis. Right? In mitosis, what happens? The 2N cell or the diploid cell, yes, divides into 2. Right? It forms the same 2 cells. Right? Same 2 cells, same 2N cells. Right? In mitosis. Right? Yes, then you have to remember what are the instances that the mitosis is going on, right? Growth of the multicellular organisms, right? Growth of the multicellular organisms and in the asexual reproduction, right? Growth of multicellular organisms. Growth of multicellular organisms and asexual reproduction. Right, in asexual reproduction, there are also the mitosis is going on, and in where the wound healing, right, wound healing, right, there are also mitosis is going on, right, because the existing cells divides and form other new cells, right, that is mitosis. Then in meiosis, what happens, Pute? Right, in meiosis, from the existing cell, yes, from the existing cells, it forms how many cells? 4 other cells. It forms 4 other cells. But first it divides into 2, right? And again it divides into other 2 cells, right? This 2N, and it divides into other 2. Then it forms 4 haploid cells. Yes, 4 haploid cells. It forms 4 other haploid cells. This happens mainly only in sexual reproduction, when making gametes, maybe in the animals or in the plants, when making pollens in plants and making gametes in the sexual reproduction in animals, right? So the meiosis going on only in when making gametes in plants and animals, right? And the importance here is, right, when we consider the mitosis, you have to remember the differences between these two, a comparison between the mitosis and meiosis, Pute, right? Here in meiosis, 2 cell divisions are there. So 2 cell divisions going on, but here in mitosis, only 1 cell division, right? Here, mitosis take place both in the haploid cells and the diploid cells. But the meiosis only take place in diploid or the 2N cells, right? Diploid cells, right? Here, in meiosis, right, the variations can occur, right? New varieties can occur in meiosis, but here in mitosis, the offsprings, or normally we say the daughter cells, are same as the parent cell, right? Here the 2 daughter cells appear in mitosis, 2 daughter cells, but here in meiosis, 4 daughter cells appear, right? 4 daughter cells appear, right? Here in mitosis, the same number of chromosomes as the parent cell, right? Same number of chromosomes appear in the daughter cells. But here in meiosis, half of the chromosomes goes to the daughter cells, right? Half of the chromosomes from the parent cell goes to the daughter cells, because from one parent cell, it forms 4 daughter cells, right? Here in the mitosis, daughter cells are same as the mother cell, because they have the same number of chromosomes, but here the daughter cells are different from the mother cell, right? So those are the differences. There's a nice table is given in your textbook, right? So draw the two sketch for the mitosis and meiosis. Just below that one, write the examples, that means where these divisions take place, and the comparison between the mitosis and meiosis, right? Most probably at least one question is given from this part, Pute, right? So it is very easy to very important to remember this one, right, about the cell division, right? Right, then we will move to the next part. Right, then the next biology lesson is the 13 lesson, Pute, in the part 2 book. 13 lesson that is about the biosphere, right? The lesson biosphere, right? There we discuss about the classification of organisms. Classification of organisms, right? Simply, right? Can you remember what is classification? Right? Yes. What do you mean by classification? Right? We group the animals. Sorry, maybe the animals or the plants, right? Group in the organisms into different levels. So we can say into different categories based on their common features, right? Group in the organisms into different levels or different categories based on their common features. That is called classification, right? Grouping of organisms. Grouping of organisms into different levels based on their common features. Based on their common features, right? You should have the idea about the classification, Pute, right? What is classification, right? Then why we classify the organisms? Maybe the animals or the plants. So we can say what is the importance of classifying organisms? What is the significance of that? Yes, it is given in the book. Yes, it is easy to remember or it is easy to study, right? When we study about the animals, when we are studying one by one, it is very hard to study, right? So we categorize the animals, or we group the animals into different sections, different levels, right? Then we go through the whole category, right? We go through the features of the whole category, then it is easy to remember, right? And if we are find a new animal or new organism according to the features of the new one, we can categorize that, right? Then it is very easy, right? Otherwise, we can't group a new one, right? But here, if we know the common features of different levels, if we find a new one according to the features of the new one, we can group that new organism to the relevant category, right? And we can find the organisms with the economical values, right? Like that, there are many significant or important things of the classification of organisms, right? You have to remember that one, Pute, right? What is the importance of classifying organisms, right? Then mainly there are two methods here. We discuss here the methods of classification. Right? The methods of classification, right? There are two methods here, right? Artificial methods, or we can say the artificial classification and the natural classification. Artificial classification and natural classification, Pute, right? In artificial classification, what is the basis? Yes, what we use to classify the organisms when we use the artificial classification, right? There we use the external features, right? In artificial classification, we use external features or their habitats, living habitats, right? But in natural classification, we use the natural relationships of among the animals or among the organisms. Natural relationships. Or we can say in natural classification, we use evolutionary relationships. Right? Evolutionary relationships. So the best one is the natural classification. Right? We cannot group the animals or group the organisms, Pute, by using the external features. Right? It is not a correct one. When we use the external features or the habitats, right? It is not acceptable. So the very good classification is the natural classification, right? There we use the natural relationships among the organisms or the evolutionary relationships, right? So by using the natural classification, all the organisms categorized under main three domains. What are the three domains? The three domain system of classification. Yes, the three domain system of classification are introduced by, yes, who is the scientist? Three domain system or three domain classification. Three domain classification by, yes, Carl Woese. Three domain classification by Carl Woese. Yes, what is it? All the organisms categorized under three domains. What are the three domains? Domain Archaea. Yes. The second one, domain Bacteria. The third one, yes, the third one, domain Eukarya. Yes, the domain Archaea, domain Bacteria, and the domain Eukarya. That is the three domain classification by Carl Woese. It is a natural classification because they are the Carl Woese used the evolutionary relationships among these organisms, right? Yes, in archaea, yes, in archaea, all the organisms are prokaryotes, right? Yes, all the organisms are prokaryotes. That means they do not have an organized nucleus. They do not have an organized nucleus in the organisms in domain archaea. That is why we consider them as prokaryotes. Yes, and the other special feature is they are not sensitive to antibiotics. They are not sensitive to antibiotics, right? All are prokaryotes and they are not sensitive to antibiotics. And you have to remember the examples, Pute, who are the organisms in archaea. Write the examples. Methanogenes, and Halophilus. Right? Those are the examples for domain archaea. Right? Then about domain bacteria, right? In domain bacteria also, the organisms are prokaryotes. That means they also do not have an organized nucleus. Prokaryotes, right? And the other feature is the bacteria, they are sensitive to antibiotics. Sensitive to antibiotics. And what are the examples? The organisms, bacteria and cyanobacteria belong to this domain. Bacteria and cyanobacteria. Yes, bacteria and cyanobacteria belong to the domain bacteria. Right? Then when we consider the domain eukarya, Pute, it again divides into how many? 4 kingdoms. 4 kingdoms. What are the 4 kingdoms? Yes, 4 kingdoms. What are the 4 kingdoms? Kingdom Protista, then Kingdom Fungi, then Kingdom Plantae, and the other one, Kingdom Animalia. Yes, those are the 4 kingdoms contained in the domain Eukarya, Pute, right? Domain Eukarya again divides into 4 kingdoms. Kingdom Protista, Fungi, Plantae, and Animalia, right? Then in the Kingdom Protista, right, we can find algae and all the protozoa belong to this one, right? Algae and protozoa, right? Like the Paramecium, Euglena, Chlamydomonas, they belong to this one, the Kingdom Protista, right? Then when we consider the fungi, they have a chitinous cell wall. You have to remember that one. The special features in the fungi kingdom, they have a chitinous cell wall. It is a very special thing, Pute, right? You have to remember that one. right? They have a chitinous cell wall, and the fungi, they can be unicellular or multicellular, and they form the symbiotic associations. That means like that fungi lives with algae, right? With uh, giving benefits to both the parties, right? Algae give a benefit to the fungi, and fungi give a benefit to the algae, right? Those are called symbiotic associations, right? Lichen is an example for that one, lichens, right? So they form symbiotic associations. That is a very special feature in fungi, right? And kingdom Plantae, you know, all the plants belong to that one, and kingdom Animalia, all the animals belong to that one, right? Separately, we have to discuss about Plantae and Animalia, right? So these are the very basic things about the classification. 3-domain classification by Carl Woese, right? Domain Archaea, bacteria, and eukarya. Then again, eukarya separates into 4 kingdoms: kingdom Protista, fungi, plantae, and animalia. Right? Then we will move to the kingdom plantae. Right? Kingdom plantae. When we consider all the plants, Pute, right? When we consider all the plants, mainly we can divide the plants into 2 groups, right? By using the feature whether they bear the flowers or not bearing the flowers, right? If they do not have flowers, we name them as non-flowering plants, right? Non-flowering plants, and the other one then flowering plants, right? Non-flowering plants and flowering plants, right? When we consider non-flowering plants, if they do not have flowers, mostly, right, they do not have seeds. But there are some plants, whether they do not bear the flowers, they have seeds, right? So non-flowering plants, again, we divide into 2 as seedless plants, right? Seedless plants and seeded plants. But these plants, they do not have flowers and do not have seeds, right? This category, whether they do not have flowers, they have seeds, right? Non-flowering seedless plants and non-flowering seeded plants. I think you can remember the examples for seedless. Yes, what are the examples? The very primitive plants that we can identify: Marcantia, yes, Marcantia, Pogonatum, what? The Drinaria, right? Like that, they do not have flowers and no seeds. Non-flowering seedless plants, right? Then there are some plants, whether they do not have flowers, they have seeds. What are the examples? Yes, Cycas and Pinus, right? Cycas and Pinus, right? The special thing here is whether these plants have seeds, Pute, right? Whether these plants have seeds, the seed is not covered with the fruit, right? Because they cannot produce the fruit because they do not have flowers. Normally, you know, after the pollination and fertilization, the flower derived into a fruit, right? In these plants, they do not have flowers, so no fruits, right? So the seeds are open to the external environment, right? External environment, the seeds are not covered with the fruit. So we name these plants as gymnosperms, right? Gymnosperms. Non-flowering seeded plants as gymnosperms. We will write that one also, gymnosperms. Right, gymnosperms. Is there, right? Right? Whether they have seeds, the seeds are not covered with the fruit. That is why we name them as gymnosperms, right? Then the flowering plants, Pute, right? Then the flowering plants, again, we can divide into two categories. I think you can remember as monocots or the monocotyledonous plants and the dicots or the dicotyledonous plants. Monocots and dicots. Monocotyledonous and dicotyledonous, right? There also we have to remember the differences between these two, monocots and dicots, right? When we consider, right, I will separate here the number of cotyledons. Wait, right, I will write some. Right, monocots and dicots. Right, I won't write all the features here, then it is easy to remember, Pute, right? Number of cotyledons. Right, number of cotyledons in the fruit or in the seed, the number of cotyledons. If it is in monocots, only one cotyledon. In dicots, there are two cotyledons in the seed, right? When we consider the stem, right, we are discussing or we are comparing the features in monocots and dicots, right? In monocots, the stem unbranched, but here in dicots, the stem is branched, right? When we consider the root system, from grade 7, you learn these things, Pute, right? From grade 7. Root system in monocots, yes. What is the root system there? Fibrous root system. And here, there's a taproot system, right? I write in short form, fibrous root system and taproot system, right? You can draw diagrams also, then it is very easy to remember, right? Then when we consider the venation, venation means the arrangement of the veins in the plant leaves, Pute. In monocots, we can find parallel venation, right? Here in dicots, we can find reticulate venation, parallel venation and reticulate venation, right? When we consider the flowers in monocots, trimerous flowers are there. Trimerous means the number of petals, 3 or multiples. Right, the number of petals, 3 or multiples, that means 3, 6, 9, 12, in that way, right? Here in dicots, 4 or 5 or multiples, right? 4 or 5 or multiples. right? And here in monocots, right, no secondary growth. Secondary growth in monocots, no secondary growth. But here in dicots, we can identify the secondary growth, right? Monocots and dicots. Monocots and dicots, right? And you have to remember some examples, Pute. For monocots, what are the examples? Paddy, right? Grass. Yes, areca nut, coconut, right? Like that, you can remember some examples for monocots. For dicots, very easy, like mango, right? Guava, jack. In that way, you can remember the examples for monocots and dicots, right? So this is all about the plants. Right? Then we move to the kingdom Animalia. Right? The next part, Pute, kingdom Animalia. Right? So for this one also, by using one paper, you can make a note. But there's a special thing, Pute, right? When we consider the groups of animals are in different the main two groups under the uh, subgroups, when we consider, there you have to remember the features, Pute. That's the very important thing, right? So it is but the useless to write all the features given in the book in a separate paper with different colors, right? To make a short note. There are all the features in each and every group, and the examples you have to remember, Pute, because that if you are given the sentence type question for the MCQ part, you have to remember each and every feature of the given groups, right? So the very important thing is, under the kingdom Animalia, you have to go through the textbook very carefully, right? Thoroughly you have to go through the textbook, Pute, and you have to remember each and every feature under the given groups, right? But here we will make a short note to remember the things, right? As you go through the short note, when you get the name of the group, right, then you have to imagine or you have to remember all the features under that group, right? For that one, we can make a short note about the animals, right? So when we consider the animals, this one also we start from grade 7, right? Animals, we divide the animals into main two groups. Yes, that is by using the feature presence or absence of a backbone. That is the criteria we use, Pute, right? Presence or absence of a backbone, or we can say the vertebral column, right? That is the feature we use to categorize the animals, right? So by using this feature, we can divide or we can categorize the animals into main two groups as, yes, what are the two groups? Invertebrates. That means they do not have a backbone, and the vertebrates. Invertebrates and vertebrates, right? Invertebrates, they do not have a backbone. Vertebrates, they have a backbone, right? Then invertebrates, again we separate into groups. Yes, what are the groups, Fute? The first one, Cnidaria. Cnidaria or Coelenterata. Next one, Annelida. Next one, Mollusca. Next one, Arthropoda. Next one, Echinodermata. Right, Echinodermata. Right, invertebrates again separates into 5 groups: Cnidaria, Annelida, Mollusca, Arthropoda, and Echinodermata. Right, so you have to remember the features of each and every group, Pute, and the examples. Right, as I told you, it is useless to write all the features here that is given in the textbook, right? You go through the textbook and remember those things and make the sketch in this way, right? Here, or at least you can write the examples, right? 2 or 3 examples, right? The animals that include in these groups, right? And what are the vertebrate groups? Yes, that means they have a backbone, vertebrate groups. Yes. His case, amphibia, reptilia, aves, and mammals. Right, these are the vertebrate groups. Yes, Pisces, amphibia, reptilia, aves, and mammalia. Right, so those are the this is the classification of animals. Right, first we divide as invertebrates and vertebrates, then the several subgroups. Right, go through the textbook and try to remember all the features in these groups and the examples, right? Because it is very important when you write the answers for the questions. Right, so that is about the animal classification, right? And in this lesson there's another uh, special thing that I have to mention, Pute, that is about the binomial nomenclature. It is a very important part in this lesson, right? Binomial nomenclature. Right, so when we write the scientific name of an organism, right, we use 2 epithets, right? We use 2 epithets to name when we naming an organism under the scientific method, right? The first one is the generic epithet, right? First one is the generic epithet, And the second part is the specific epithet. Right? So when we name an animal or a plant, we have to use this scientific nomenclature or the by we use here. Here we use two epithets. That is why we name this method as binomial nomenclature. By means to pute. Right? We use two epithets to name an animal or a plant. Right? Here we have to use some rules. Right? When we write the name of an organism using the scientific method, we use the English terms. The first letter is a capital letter, right? As an example, the first letter is a capital one and all the other are simple letters. And in the second epithet also it starts with a simple one and all the other letters are simple, right? And when we write in handwriting, we write the name in this way and we have to underline it, right? In handwriting, if we use the handwriting, right? If it is in printed form, right? It starts with the capital letter and the letters should be in the italic form, right? It should be in the italic form in when we print the name, right? So those are the things that we have to consider. Start with the capital letter and all the other letters are simple. If it is in handwriting, after writing the name, we have to underline it. If it is in printed form, the letters, it should be in the italic form, right? Those, when we use the binomial nomenclature, we have to obey those rules, right? And the two epithets, generic epithet and the specific epithet, right? So that is about the binomial nomenclature that you have to remember that part today, right? So then the next lesson, the 14th lesson, right? In 14th lesson also, I will give a rough sketch, right? That is the continuity of life. Continuity of life, right? Continuity of life. There, first we discuss about the reproduction. Yes, what do you mean by reproduction? Simply how we can explain the term reproduction. Yes, that is a life process, or that is a biological process. Yes, that gives rise to a what? New generation. It is a biological process or a life process that gives rise to a new generation, right? When we consider the reproduction, it is in two ways: asexual reproduction and sexual reproduction, right? Asexual reproduction and sexual reproduction. Mostly, the asexual reproduction occurs by spores, right? Spores or by vegetative parts. By using spores or by using vegetative parts, that means the growing parts of a plant, right? And here the sexual reproduction occurs by the fusion of gametes, right? Asexual reproduction by the spores or by the vegetative parts, and the sexual reproduction by the fusion of gametes, right? And here, that for this one also, you can make a comparison between the asexual and the sexual reproduction, right? A nice comparison is given in the book. Here for the asexual reproduction, only one parent is contributing, but here for the sexual reproduction, two parents should contribute, right? Here in asexual reproduction, the offsprings or the second generation is same as the parent generation or same as the parents, but here in sexual reproduction, variations can occur, right? Variations can occur, right? And here in asexual reproduction, no gametes are produced. Here in sexual reproduction, gametes are produced. Here only mitosis occur. In sexual reproduction, both the mitosis and the meiosis occur, right? Meiosis should be there to form the gametes, right? So here the new variations are not formed, but here in sexual reproduction, variations occur, right? Like that, we can make a comparison between the asexual reproduction and sexual reproduction, right? Then we will move to the plant reproduction. Right? Reproduction of plants. Right? In plants also, we can identify both the asexual reproduction and the sexual reproduction. Right? Asexual reproduction and the sexual reproduction. In asexual reproduction, again, there are two types: natural asexual reproduction and artificial asexual reproduction. Natural and artificial, right? Natural and artificial. Natural reproduction occurs in different ways, right? That means how they form the next generation, right? How they form the next plants, right? By roots, right? These are the methods that naturally, right? How the plants make their next generation, right? By roots, by leaves, by suckers, by runners, right? You have to write the examples here also, right? Leaves, by suckers, by runners, then by bulbils, and by underground stem. Right? By underground stems. Those are the natural asexual methods that plants use to make their next generation. Right? When we consider the underground stem, again, there are four types. Right? Rhizome, comb, bulbs, and stem tubers. Right? For each and every one, you have to write the examples here. What are the plants, right, that form new plants by roots? Are like curry leaves, breadfruit, like that. Then what are the plants that form the next generation or the new plants by leaves like Akapana, Begonia? Then what are the plants by suckers? Then runners like Gotukola, sweet potato, then bulbils, then the underground stem. Right? Here underground stem, you know the underground stem means the stem is just below the ground. Right? So they do the using the underground stem, they do the vegetative reproduction. That means they form the new generation, and also during the unfavorable conditions they can survive, right? Because the stem is just below the ground, right? Rhizome, in rhizome pute, the stem is, now I told you the stem is just below the ground, right? Here in rhizome the stem is parallel to the ground. This is the stem, right? Like in ginger, turmeric, right? The stem is parallel to the ground. But in comb, the stem is, right? The stem is in this way, right? In the vertical position. It is perpendicular to the ground, right? In bulbs means like in onions, bulbs, right? Then the stem tubers as in the potatoes, right? Stem tubers, right? Rhizome, the underground stem is parallel to the ground, right? In comb, the underground stem is perpendicular to the ground, right? Anyway, the stem is just below the ground, right? So that is about the natural asexual methods, right? You can mention the examples here when you make the short note, right? Then the artificial methods, Pute, right? Artificial methods of asexual reproduction. That means with the involvement of the man, we do the asexual reproduction, right? By using a growing part or a vegetative part of a plant, we get a new plant, right? Artificial asexual reproduction, right? What are the methods? Rooting of stem cutting, right? Rooting of stem cutting. That means we cut a part of the stem and we grow it. We grow it as a new plant, like in roses or the shoe flowers, like that, right? Then the next one, grafting, right? Grafting. There are two methods. What is bud grafting and twig grafting, right? And layering, right? Layering also in two types, but the ground layering and air layering, right? Ground layering and air layering. And the next one, tissue culture, right? Tissue culture, right? Tissue culture is a method that we can get more new plants, right? With a short period of time, that have the same features as the mother plant, right? But here you have to remember, Pute, in tissue culture, we have to use the controlled environmental conditions. Most probably it is done inside the labs, right? Tissue culture, right? It is somewhat a developed one, but these three, right, very easily we can do, right? If the natural asexual method not occurring, we have to use these artificial asexual methods, right, to get the next generation, right? Then when we consider the sexual reproduction of plants, what is the reproductive organ in plants, Pute? That is the flower, right? In flowers, the pollination occurs, right? Pollination occurs, right? Pollination is in two types. Yes, the self-pollination and the cross-pollination. Self-pollination and the cross-pollination. What is pollination, Pute? Deposition of the pollens on the stigma of the flower, right? This can happen in the same flower or in a different flower, right? If the pollen deposit on the same flower stigma, that is self-pollination. If the pollens deposit on a stigma of another flower, that is cross-pollination, right? So in your book, you are given all these things about the pollination. What are the agents of pollination? Then how the pollination occurs, then the fertilization, then the appearing of fruit, then how the fruits are dispersed by water, wind, by animals, and by the explosive mechanism. What are the adaptations that plants show, right? Like that, and the human reproduction, right? The two reproductive systems in male and female go through the parts and the functions and the menstrual cycle and the childbirth, right? So go through these things in the lesson that is given in the book, right? We will move to the uh, paper, Pute, right? Just I will uh, give the answers, right? Because I think that uh, you have go through the paper and write the answers, right? Right? This is the paper, right? I will tell the answers, Pute, right? For the first one, what is the biological molecule out of the following? That should be lipid, right? Second answer. Second one, select the answer containing an organic compound and an inorganic compound consequently. The second answer, liquids and minerals, right? What is third one? What is the compound contain nitrogen and phosphorus? Fourth answer, nucleic acid, right? Then fourth one, what is the incorrect about lipids? It should be the fourth answer. Hydrogen and oxygen proportion in lipids is 2 to 1. That is the incorrect one. Fifth one, what is the special protein in red blood cells? Hemoglobin. Normally it should be globin. The protein should be globin. When it combined with the iron part heme, we name it as hemoglobin, right? Sixth one, what is the monosaccharide from the following? What is the monosaccharide? It should be glucose. Glucose is the monosaccharide, right? The seventh one, the element needed to produce hemoglobin is iron. Fourth answer, it should be iron, right? Question number 8, a monosaccharide without sweet taste, that should be galactose. Galactose. The ninth one, deficiency of which element causes chlorosis in veins? You have to go through the deficiency symptoms, Pute, right? In areas and between the veins, that is sulfur. First answer, sulfur. Question number 10, the incorrect statement on virus, DNA and RNA are present. Not both present in virus. Only DNA or RNA present in virus. DNA or RNA, not both, right? Question number 11. A nucleotide is made by the combination of several components. Which one is not a component? Not a component, that is nucleic acid, right? Question number 12. Which reagent is used to test for starch? That is iodine. The reagent we use to test for starch. Question number 13, right? The organelles that we can find in and plant cells, that is A and B, cell wall and the central vacuole, right? Question number 14, only to plants, A, C, D, only A, C, D, third answer. Question number 14, third answer. Question number 15, B only, the mitochondria, third answer, B only, right? 16, the most essential organelle that should be present in typical cells to pass the hereditary characters, that should be nucleus, because the chromosomes are there inside the nucleus, right? Nucleus. 17, in which of the following instances does meiotic division occurs? Meiotic division occurs when gametes are formed during the production of ova and sperms. Ova and sperms. Right? Fourth answer. Right? Question number 18. Right? A cell division is given. We have to find what is A and B. Right? A is meiosis and B is mitosis. Fourth answer. Meiosis and mitosis. 19. Which of the following is a non-living structure? That should be cell wall. Second answer, non-living structure. What is the cell organelle without a membrane? That is ribosome. Without a membrane, ribosome. What is the cell organelle maintain the water balance? That should be vacuole. Fourth answer. 21, fourth answer. 22, ribosomes are attached to which part? Right? Not the, it should be endoplasmic reticulum. If the ribosomes are attached, we name it as rough endoplasmic reticulum. Rough endoplasmic reticulum. Right? Here you are given a figure of a cell. Right? Question number 23. What is the letter denoting the control center? Control center means the nucleus, Pute. Right? Here the nucleus denoted by the letter I. Right? I, third answer. What is the letter denoting the organelle where photosynthesis is done? Photosynthesis is done. That is in chloroplast, letter G. Letter G. Right? Then select the multicellular organism. Chlamydomonas, Euglena, Paramecium, all are protozoa and all are unicellular organisms. Chlamydomonas, Euglena, Paramecium. Right? And the multicellular one is Hydra. In the Cnidaria group, Hydra. Right? 26. The organizational levels are given. Cell, then tissue, then organ, then system, then organism. Then A should be tissue and B should be system. First answer, tissue and system. Right? Then some substances removed from living organisms are given here. What are the excretory substances? Sweat, inhaled, exhaled air and urine. A, B and C. Feces is not an excretory substance. You have to remember that one. Feces means the undigested food, right? And it is not produced in the metabolic activities, right? Normally, the we consider as the excretory substances the waste products that form during the metabolic activities, right? So, sweat, inhale, exhaled air and urine are the excretory substances, right? Then question number 28. What is the single cell that builds up a new organism? The single cell, that should be zygote, right? With the combination of the ovum and the sperm, the single cell produced is zygote, right? Then 29, in which process given below does the number of cells increase irreversibly? That is growth. The irreversible increasement of the size of the cell or the dry mass, that is growth. A virus is made of nucleic acid and a protein sheath. No any other organelles are there in a virus. Only the nucleic acid, that means DNA or RNA and the protein sheath, right? The instrument is given here, the axonometer. Axonometer, right? What is the life process that we observe using this one? That is growth of plants, right? Growth, axonometer. 32, what is the byproduct of photosynthesis? It should be oxygen byproduct. The main product is glucose and the byproduct is oxygen, right? Then question number 33. What is the group of microorganism having cell wall of chitin? Having cell wall of chitin, we discuss it, fungi, right? They have a chitinous cell wall. A characteristic of bacteria, a cell wall is present. Fourth one, cell wall is present. 35. Which is the phylum with highest number of species in the animal kingdom? Yes, that is Arthropoda. 75% of the animals belong to this one, Arthropoda. Right? 36. Which of the following is not a feature of all chordates? Not a feature of all chordates or the vertebrates. You are given the diagram also. Right? Presence of post-anal tail is there. Right? And the presence of spinal cord and the ventral heart and no lungs. Presence of lungs, that is not the feature of all chordates. Right? Second answer. 37, which is not a use of classification of organism, right? Second one, having a knowledge about the number of species, right? Number of species. It is not a use of classification. 38, which is the invertebrate group not having a coelom, that is the first group, the coelenterate, right? They do not have a coelom, only they have a cavity, right? Coelom, coelenterate group, right? Then 39, what is the invertebrate phylum that shows the evolutionary relationship to the phylum chordate, the invertebrate group, the last one, echinodermates, right? Echinodermata, they have the evolutionary relationships with the chordates. 40, common feature of aves and mammals, right? Aves and mammals, this second one at the fourth one, both are correct, Pute. Homeothermic and breathing by lungs. Homeothermic, breathing by lungs. 41. Which of the following represent a stage in the sexual reproduction of plants? Sexual reproduction, obtaining a young plant from a seed. Sexual reproduction means getting new plants by the seeds, right? So the first answer. 42. Monoecious plants are those bear both the female flowers and male flowers. Monoecious. Both the male and the female flowers are in the same plant. Monoecious. Right. 43. A special adaptation found in flowers for cross-pollination. Cross-pollination, that means the deposition of pollens on a stigma of another plant, right? Plants have some adaptations to avoid self-pollination and to promote cross-pollination, right? What is the adaptation of this one? Androecium or the gynoecium mature before the other, right? Or the androecium or the gynoecium mature in two different times, right? So the answer should be the fourth one, right? So those are the answers for the paper, Pute, right? I think you got the idea how you have to make a short note or how you have to study, right? But you have to remember, you have to go through all the lessons, right? You have to go through all the lessons because as I told you, from each and every sentence given in the textbook, right, they can make a question, right? So go through all the biology lessons, right? Then make the short notes, right? Then we will stop for today, right? Good night, all of you.